Okay, so we are now live. Good afternoon, everybody. It's really a pleasure to have all you guys here. So we are being live streamed on YouTube at ICB, USP, at the Biophysics Institute, and through these Zoom room all over Brazil. Once again, it's another installment of the Immuno Webinars conference series that is organized by Gustavo and Diego at USP, by me, Bruno Dias, Cristiane Bandeira de Mello, and Miriam Werneck at Federal University of Rio de Janeiro, and by Dr. Thiago Mineo at Federal University of Uberlândia. And today we have the pleasure of hosting Dr. Thomas Kaufmann, who holds bachelor's and doctorate degrees from the University of Freiburg, followed by a postdoctoral fellowship at the Walter and Eliza Hall Institute of Medical Research in Melbourne, Australia, under Andreas Strasser. Since 2013, he has been an associate professor at an Institute of Pharmacology at the University of Bern in Switzerland, where he leads the laboratory of programmed cell death, which is a subject that he made extensive and really important contributions throughout the years. Today, he's going to talk about a different aspect of his work, focused mainly on basophils and mast cells and their applications in allergy biology and diagnosis. So thank you again, Dr. Kaufmann. You have the floor.
Well, thank you very much, Bruno, for the kind introduction. I hope you can hear me well, everybody.
Yes.
So good afternoon from Switzerland. I would have loved to come in person to Brazil. Never been to Brazil, but I hope there will be an opportunity once in the future. But I'm very excited to talk to you here on this Immuno webinars online. And so thanks a lot for giving me the opportunity to talk about more recent development in my lab. So I will not talk about cell death at all, which is a bit of a new thing for me. And because I'm talking about our allergy project, I have to start with a short disclaimer. So I am co-founder, board member, and scientific advisor of a spin-off company of the University of Bern called Atanis Biotech, which is offering services in the space of allergy diagnostics and research. Good. Just as a little introduction, what our lab usually does, or my lab. So I'm a cell death person by origin. So for the last 25 years, I've been focusing on programmed cell death, a lot on apoptosis, especially BCL2 proteins. Get the laser pointer. In health and disease, and that's mostly cancer, application of novel class of drugs called Ph3 mimetics in cancer, and non-apoptotic roles of BCL2 family members, as well as more inflammatory forms of cell death, especially necroptosis. And when we started on this project, we started to work on innate immune cells, including granulocytes, and from there, things evolved, and I tried to explain you in a bit how then we came about to working with basophils and mast cells and starting to work on actually allergy signaling, which is a relatively small part of my lab still, but it's growing, and I'm going to focus on this third subject of interest today. Bit of issues with okay. So I'm going to mention HOXB8 a lot today, and HOXB8 is a homeobox gene that for a long time has been known to be able to immortalize myeloid progenitor cells, especially from a murine system. So this works much better in the mouse than in human cells, but by overexpressing HOXB8, and it's also true for HOXA9, another homeobox gene. So these are transcription factors. They block differentiation of myeloid progenitor cells, and they also block apoptosis. That's also how we got interested in these genes originally. And this, as I said, has been known for a long time. It was in 2006 then when the group of Mark Camps and Hans Hecker published this, what I consider a landmark paper, where they showed very elegantly that by introducing conditional HOXP8, so a HOXP8 that can be switched on or switched off, they could generate or they have developed a technology to really produce in vitro differentiated macrophages or neutrophils in large quantities. And so really the trick is to conditionally immortalize hematopoietic progenitors isolated from mouse bone marrow and in the presence of the right cytokine mix. So this is very important, and I'll come to that. And then immortalize the cells and then switching HOXP8 off again and then force the cells to differentiate in vitro into mature neutrophils or macrophages. So in this paper, they mostly showed it for neutrophils and macrophages, where they can really get a very pure population of neutrophils within just a few days, actually, of differentiation. And in the paper, they also described variations to generate macrophages. So this really was the start. And they and others over the last 15 years have really developed this technology much further. And nowadays, it's using this idea or this technology, almost all hematopoietic lineages can be generated with this conditional HOXP8 or HOX gene step, including lymphocytes, which are not shown here on this cartoon, but all myeloid lineages, but also lymphocytes with some variation of the protocol. And so what they originally, so what they use a lot is a fusion protein between the estrogen receptor and the HOXP8 gene or HOXA9. So this fusion that is present on an expression cassette that can be switched on or off. And what's critical is the cytokines that are present during the immortalization. You end up with immortalized cell lines that behave almost, like a cell line that can be maintained and cultured pretty much forever. And then by switching off the homeobox genes, or by removing the estrogen in this case, and adding the right selection of cytokines, again, you can drive or force differentiation along a specific lineage and produce pure populations of mature and differentiated cells listed here. And this usually only takes 5 to 6 days, so it's a very fast process. It's also not that clearly understood why it only takes a couple of days for some lineages, and come to that later. So when we got interested in neutrophils, that was around 2008, 2009, when I started as a junior group leader here at the Institute of Pharmacology in Bern. And we started to use this conditional HOXP8 system with the same principle, but slightly different tools. So we had access to different tools. I don't think the differences are hugely important, but there are some differences. So we don't use an estrogen receptor HOXP8 fusion gene, but rather we regulate the expression of wild type HOXP8. So we use a different expression system, and we don't use retroviruses, but lentiviruses to transduce the cells. And we have adapted the system to work in a very cost-efficient RPMI medium. And there's other minor changes. I don't want to go into too many details, but this worked pretty well straight away. There's an example here of neutrophil lineages. So we have this neutrophil progenitor lineages, these immortalized cells, and we can shut down the HOXP8 again. So by removing tamoxifen. So our expression system is tamoxifen inducible. You see here the exogenous HOXP8 expression of the progenitors. We wash the cells, remove the tamoxifen, and within 5-6 days, we go from progenitors to differentiated mouse neutrophils. And we and others have shown that these neutrophils resemble pretty well primary neutrophils isolated from the bone marrow of mice, so mature but not very mature neutrophils, and you can further maturate them by adding GCSF, for example, for another day. And we use this system extensively. So here's some characterization of typical surface markers, CD11b, GR1, for example, become highly positive. The cells lose c-KIT expression when they differentiate, so the progenitors express c-KIT, the neutrophils don't express c-KIT. And we take the neutrophils in culture and they die with the similar kinetics as bone marrow and isolated neutrophils, primary neutrophils, and this is strongly blocked by adding GM-CSF or caspase inhibitors. So we use the system a lot to also to save the need to sacrifice mice, because at least for the cell death research that we did, these Hox-based neutrophils turn out to be very, very useful. And just to give you an example, really the quantities you can produce with this Hox-based system is shown here. So I'm not going to explain what we really did here. So we looked into different cell death signaling pathways, but the point is these are Western blots of 20 conditions. And so for this experiment, neutrophils, you use a lot of neutrophils for protein extracts. So they don't have a lot of proteins compared to other cells. So for a Western blot extract, we typically use 5 million neutrophils for one lane or per condition. So for this experiment shown here, we used 100 million neutrophils, Hox-behave neutrophils, and that's very easily achievable in 200 milliliters of RPM-I-based differentiation medium within, as I said, 5 days from the progenitors that we had. And so we use this system a lot to do biochemical analysis, where you use a lot of material. And if you wanted to do that with primary neutrophils isolated from mice, for such an experiment, you would have to sacrifice at least 10 to 15 mice for this Western blot. So I think there's also a big ethical motivation or 3R aspects that we can use the system to save mice, actually. So these are neutrophils. What about basophils? Then, at the time, 2010, 2011, we thought, would it be possible to use this Hox-based system to generate basophils? Because basophils, the situation is much worse than for the neutrophils. So you can isolate basophils from mice, primary, but, very few. Basophils are very rare white blood cells, and it's usually less than 1% of peripheral blood, or 0.5 to 1%. So the green cell here would be basophil, one in 100 roughly. And I thought, so it would even be, very exciting if we managed to make the system work to in vitro differentiate basophils. And so a PhD student at the time, Ursina Gurzeler, spent a lot of her PhD trying to generate basophils from hematopoietic progenitor cells, so from bone marrow of mice. And really the trick here is to use the right cytokine. I guess all of you know, or most of you know, that to generate basophils from bone marrow, you need interleukin-3. And so this is the biggest change to the protocols that existed before, is the switch to IL-3 before immortalization, during immortalization, and then also during the differentiation. So we managed to generate so-called IL-3 dependent conditional Hox-behave progenitor cell lines that can then be maintained for very long times in the presence of tamoxifen. So when Hox-behave is switched on, they maintain their progenitor stage, and then when we wash the tamoxifen away, we get basophils, mouse basophils, within 5 to 6 days. And that's actually very similar to what you do when you differentiate mouse bone marrows in IL-3 to basophils or mast cells. It's just this intermediate step of immortalization. And then again, from the progenitors to the basophils, it only takes 5 to 6 days. And when you want to do that without the Hogsmeade immortalization, it takes much longer, and it's not very clear why this is the case. And so here, again, that's our first publication to show that we can generate basophils, again, upon shutdown of HOXB8, and cells differentiate from progenitor to mature basophils, very high in the Fc epsilon receptor 1 alpha chain, so the high affinity IgE receptor, which is a typical marker for basophils, as well as usually we test for c-KIT and Fc epsilon receptor 1 alpha. And the basophils really are positive for the Fc epsilon receptor, but negative for c-KIT. Whereas bone marrow-derived mast cells that we used here as a control are very high in c-KIT expression also, so they are double positive for IgE receptor and c-KIT receptor. So we did other characterization, and one is shown here. So there's a set of specific proteases called mouse mast cell proteases. Despite their name, some of these mast cell proteases are more specifically expressed in basophils, and other are more specifically expressed in mast cells. And so the mast cell protease 8 and 11 are pretty good markers for basophils, whereas the mast cell protease 4 and 5 are usually markers for mast cells. And as is shown here, the Hogsmeade basophils, as we call them, express the right set of mast cell proteases. And so that was more than 10 years ago. At the time, we had no really idea or plan that this could be used for allergy research. So we had no projects ongoing. We were like cell death people. And what we actually did in a couple of publications and follow-up protocols, projects, was to investigate cell death pathways in these basophils. And one application that we think could be interesting is this new class of drugs called BH3 mimetics. So those of you who work a bit on cell death certainly know about the BH3 mimetics. So these are drugs antagonizing the anti-apoptotic BCL2 family members very specifically, and are exclusively used or have been designed to treat cancer. At the moment, it's more hematological cancers, but there's a lot of interest. And so we hypothesized that these drugs could be rederived for other indications, including immune disorders where you have overactivated immune cells, such as allergic effector cells, such as basophils or mast cells, or activated neutrophils. And we did some really, preclinical work to show that this can work, or there's limitations, but there's some specificity to kill selective lineages with these drugs, and that's how when we started to think about allergy and allergic effector cells. So that's what I'm going to talk about now. And it's clear that interleukin-3 is not only a key cytokine to differentiate basophils from bone marrow progenitors, but of course also mast cells. And those of you who work with mast cells or with mouse mast cells, you take mouse bone marrow, put them in culture with IL-3, and after 3 to 4 weeks, you end up with a nice homogeneous population of mast cells, so the classical bone marrow-derived mast cell protocol. What you also get after about 10 days are basophils, but they are much shorter lived and they will die off And so after 3-4 weeks, you end up with mast cells. And the reason is that the mast cells and the basophils, you know, they're quite close together in this differentiation tree here. You know, they have common progenitors, basophil mast cell common progenitors, and they split up into basophil progenitors and mast cell progenitors. But there are many similarities between these two cell types. So having said that, we were not really surprised that when we generated this IL-3 dependent HOXB8 cell lines from mice, you know, we generate many lines, and actually not a few lines look like this. So they didn't look like basophils, they didn't look like you know, Fc epsilon receptor positive, c-KIT negative only, but they look like a mix of at least two populations, as shown here, and we hypothesized at the time that the lower population here might be basophil progenitors, and the upper, the c-KIT positive, might actually be mast cell progenitors. And so what we did quite recently, actually, and that was also after, you know, we had exchange with Bruno Diaz when he was in Germany for his research stay, made us think a bit, you know, we should really get into subcloning these mixed lineages and see what we get. And so this is very recent work, actually, and quite surprisingly is that when we get nice subclones from such a mixed line, we end up again with a good percentage of mixed lines. So we do end up with basophils, basophil committed lines. We end up with mast cell committed lines. And this again is characterized by this specific mast cell proteases, as shown here. This is now done by the qPCR. So there's clear evidence that we can get mast cell progenitors out here, or basophil committed progenitor out here, but there also seems to be a common mast cell basophil progenitor in here. So it's still not clear exactly at which stage this HOXB8 immortalizes the progenitors, or what the plasticity is of this process. And so here we're planning to go deeper into this using, you know, single cell RNA sequencing to really figure out what makes a progenitor become a mast cell, what makes a progenitor become a basophil. And I think this HOXB8 system is a beautiful system to study this kind of relationship between mast cells and basophils in the mouse. So this, I mentioned it before, but just to highlight, this HOXB8 system, to my knowledge, only works in the mouse. It has been tried in humans. We have tried it in humans, and it didn't, well, we couldn't manage to generate cell lines using the same protocol. But in mouse, it works beautifully. Irrespective of whether we have basophils or mast cell committed lines, when we differentiate the cells and then look for IgE-dependent activation, degranulation, they perform very similarly, and they perform extremely well. So this is an allergen IgE-dependent activation of either basophils or mast cells, and with really nice activation curves. And that brings me to actually the allergy. The time, as you know, allergies are a huge problem, and including Brazil. There's increasing prevalence all over the world, especially in developed countries. So roughly 1/3 of the population has one or the other form of allergy. And there's massive numbers of people suffering from airway, allergic airway inflammation, including asthma, and increasing numbers of food allergic patients. So this is a big problem, and it's getting worse over time. So most allergies are the so-called type 1 or immediate response type allergy, which is mediated by IgE. And I don't think I have to explain you to this audience how an IgE-dependent allergy is developing. Just very briefly, you know, you have to get sensitized with an allergen that usually is not a harmful molecule or substance, but when you get sensitized, it can lead to allergy. So it has to enter the system. There needs to be a barrier breach of some form. The immune system processes the allergen, presents it to T cells, and then depending on the patient, of course, many factors come into play, and crucially, depending on the cytokine milieu that is present, the T helper cells then instruct B cells to produce antibody of the IgE type. So there's a class switch driven by type 2 cytokines. And when the B cells or the plasma cells start to make allergen-specific IgE, then you can become allergic to this allergen. You don't have to, but then you're sensitized to the allergen. And then the IgE binds to the Fc epsilon receptor, the high affinity receptor that sits on mast cells in the tissue or basophils in the blood. And the IgEs bind to the receptor and stay there at the surface. So they're not internalized or not very rapidly, and the cells are like covered in IgE. And then you need a challenge, a second contact with the same allergen or a cross-reactive allergen. That then leads to cross-linking of the IgE receptors, activation of intracellular signaling pathway, and degranulation of the mast cells or the basophils. And then once they degranulate, then you develop the allergic symptoms from mild to death, lethal, potentially lethal, so the whole bandwidth, depending on how susceptible you are. But of course, this is due to the release of mediators that are either stored in the granules, like histamine or other cytokines, or de novo synthesized mediators such as leukotrienes or prostaglandins. And they cause all the clinical symptoms of an allergic reaction, including itching and sneezing and short breath shortness, etc. And this is called immediate type, as this happens within minutes. So this is an immediate reaction, and you have to, react very fast if this happens to you. Good. So how is allergy usually diagnosed? And so the best tests in terms of accuracy are actually in vivo tests, the gold standard being the so-called skin prick test, which really, depending on the allergen, works more or less well to not very well at all. There's a big problem of false positive results, and especially it doesn't work very well for food allergens. The reason is that the mast cells in the skin are quite different from the mast cells in the mucosa in the gut. But it's a gold standard. There's a certain risk. So it's cumbersome, it takes time, it's painful, or often done in children. They don't like to do that. And there's a risk that the allergen can, if it reaches the body systemically, it can cause a severe side effect in the worst case. But this is still the gold standard, whereas for food allergies, the most accurate test actually is the so-called oral food challenge, OFC. As I said, the skin prick doesn't work for most food allergens. And so oral food challenge is nothing else than giving, you go to the doctor and you get a small quantity of the allergen, so in this case, a peanut to eat, and then the doctor observes if and how fast you react. And of course, this is a very, well, from the accuracy point of view, as good as it gets, but it's also the most risky kind of test you can do, so especially if you think of severe types of allergy like peanut. You know, this is very stressful for the mostly children who do this, and the parents and the doctors. Most doctors don't like to do it or feel challenged because of the inherent risk of developing severe reaction up to anaphylaxis. So the question we asked at the time, so can there be functional tests, in vitro tests, that could complement or in the best scenario replace the in vivo tests? And this was mostly initiated, or the idea that our Hogsmeade system could be used for these applications came from my colleague and close collaborator, Alexander Eckel here in Bern. So he's really the allergy researcher, has always been. And he approached me after we published our Basophil paper and said, Thomas, listen, I have an idea, or we should follow up on the idea to develop an in vitro allergy test, a functional test using your technology of Hogsmeade immortalization. And so that's how this started. And so we developed a test that we call FAST-PACE. That stands for Functional Allergy Screening Test by Passive Sensitization. And the big advantage is it's a standardized test. So a functional test, it's easy in logistics because it works with serum. That allows for pro and retrospective analysis and allows for high throughput possibility. And so what we did is we generated IL-3 immortalized Hogsmeade cell lines, not from wild-type mice, but from a transgenic mouse expressing the human high-affinity IgE receptor, so the human Fc epsilon receptor alpha chain. So we isolated lineage-depleted progenitors from bone marrow and primed them or incubated them in differentiation medium with interleukin-3, infected them with a conditional Hogsmeade virus. There's a selection step, which is not shown here. This was an antibiotic out of here. And then we generated many different cell lines, and then we characterized those cell lines. And again, once you have these cell lines, you can then trigger the differentiation in vitro on demand. And so we generated many lines of these mice. So these transgenic mice, so these are pure transgenic knock-in mice. So instead of the mouse receptor, they express the human IgE receptor and none of the mouse receptor anymore, the alpha chain. And these mice were generated by Jean-Pierre Quinet at the time at Harvard University. So of the many lines we generated, we got the full picture in our cells that looked more like basophils, cells that looked more like mast cells, and many mixed cell lines. And so we tested them and chose the lines that performed the best in the functional assay. And this turned out to be mast cell committed cell lines. So we characterized these cells, progenitors on the left side, differentiated cells on the right side. So they strongly increased c-KIT expression. They are already high in IgE receptor, but it further increases, and it increases in granularity. So these are image stream photos. And when you look at the morphology, they really go from a cell that looks like a progenitor and is still proliferating a lot to cells that look like mast cells. And you can see these granuli here in purple. And actually, these are more or less end-differentiated mast cells, so they don't proliferate anymore after 6 days of differentiation. And so I have to speed up a bit. So we could show that these cells bind IgE on their surface in a dose-dependent manner. Actually, we can use these Hogsmeade mast cells. We could use them to quantify IgE levels in serum. So this is just a correlation of when we use our cells to determine the IgE versus the standard, which is immunocapped. But that's not the main application, of course, but still interesting. And then how we measure the degranulation of these mast cells. So we do that by flow cytometry. So once the mast cells are activated and the granulae fuse with the cell membrane, certain lysosomal associated membrane proteins called LAMPs are exposed at the cell surface, through this fusion of the granule membrane with the plasma membrane. And you can use this marker, LAMP1 or LAMP3. This is often used, also CD63. And we use LAMP1, which is CD107A, but the principle is the same. And you can easily quantify degranulation by flow cytometry. And here we have shown actually that there's a one-to-one correlation with the dextrohexosaminidase release assay, which is a standard assay to quantify mast cell degranulation, but it takes much longer and it's much more cumbersome, whereas the fax-based assay just takes 20 minutes to quantify. And so it really works nicely. So we can load the mast cells with an IgE. So here is increasing amounts of a NIP-specific IgE monoclonal antibody, and then challenge the cells with the allergen, which is NIP-BSA, and the more IgE you have on the cells, the more degranulation you get upon allergen challenge. So the positive population here are the CD107 plus positive cells, means percentage of mast cells that have degranulated. And then you can plot this activation on a graph here. So you have your IgE concentration and degranulation in percentage, and you get this signaled activation curves, and showing that our hogsbait mast cells have a remarkable potential to degranulate. So they almost 100% of degranulation can be reached. So this is one important readout. Where is this plateau? What percentage? And the other readout is the left or right shift of this curve, which corresponds with the sensitivity towards the allergen. Of course, you can also, we don't usually titrate in the IgE antibody, but rather the allergen. And so how this assay typically looks like, it's a form of a mast cell activation test. So we mimic the allergic reaction in a test tube. That's how we promote the whole thing. So what we do, or what can be done, you take a patient sample, you isolate the serum, or you prepare the serum from the patient, including everything that is in the serum. So not only the IgE antibodies, but also IgGs, allergen-specific IgGs, other things in the serum. So it's all there. You incubate this with your differentiated hogsbait mast cells. So these are still naked, so they haven't seen IgE yet. This is called passive sensitization. That is typically done overnight. The IgE binds to the cells. And then you can test whatever allergen you want, throw it onto the cells, and measure degranulation to see whether your mast cells react with this patient sample and the allergens you want to test. And you get these activation curves, as I showed you before. So here we would have a fixed concentration of serum IgE and titrate in the allergen, but the curve you get looks pretty similar, actually. And so these are now real patient data, pretty old already. So we have actually much newer data that looks even better, but just to show that this works with real patient sera. So there's 3 different patients, and all of them are positive here for peanut. And each patient reacts differently in terms of plateau that is reached, which could be a measure for severity of the symptoms. And the shift to the left or to the right would be a readout for sensitivity to that allergen. And so this, in principle, works for all IgE-dependent allergies. So there's more than 300 allergens known, and from the principle, so these are all IgE mediated. This assay should work for all IgE mediated allergies. We haven't tested all of them, but we're increasing validating this test, and here are just a couple of different classes shown: so cat allergens, venoms like wasp, bee, hostess mite, and pollen, and peanut. And we have much more data now on food allergens, and it's always working nicely in our assay. Very recently, we have contributed to a clinical utility study in peanut allergic patients. So it's really a cohort of 69 allergic patients and 27 non-allergic controls. And that was collected and analyzed by another, by the Monas group years ago. And we got access to these samples retrospectively and analyzed them in our assay, and analyzing these 100 samples is really fast to do in this assay. So it takes 2 or 3 days to run all these samples, and it's quite remarkable how well this works here with this really cohort. And so we could pick up really the allergic patients in the assay here in red, and the non-allergic didn't respond. With one exception here, there was one patient in the non-allergic group that reacted, but it turned out that this non-allergic patient had very high titers of peanut-specific IgE, so may have been a misdiagnosed peanut allergic patient. But no test is perfect. We calculated a diagnostic accuracy of the Hogsgate MAT of about 95% for peanuts, and this is extremely very excited about this accuracy. So this is really outperforming specific IgE measurements as pinprick test, and it's performing as well as the basophil activation test that is done in fresh blood from patients. So the basophils from fresh blood, and that this test that works really well, but is logistically a bit of a nightmare. So you have to draw the blood and do the test immediately because it's fresh blood. And whereas in our test, as I said, it works with serum that can be frozen and shipped around, and it's much more easy to collect samples and do the assay of hundreds of samples together. And the other advantage of the Hogsgate MAT over the BAT is that we showed in this paper here that we were able to pick up the so-called non-responders. So about 10 to 15% of allergic patients do not respond in the BAT assay because the basophils don't degranulate. And so these are called non-responders, and these non-responders are typically excluded from some such clinical studies that show the efficacy of the BAT assay. So good. Okay. How much time do I still have?
Another 10 minutes?
Is that okay, 10 minutes? Yes, that would be fantastic.
Sure.
Yeah, good. Thank you. One big application that we also see for the Hogsgate MAT is to predict immunotherapy successes. So when you treat allergic patients with increasing doses of allergen, they can become tolerant to the allergen. And so this we can see in our assay. So we have these are patients that have been treated with immunotherapy, and we collect samples, blood samples after 3, 9, 12 months, 2 years, 3 years, and then you do the assay with all these samples together in one go, and you can really see that the curve comes down to basically 0 to a flat curve, and that's when this therapy works well. And I think this is a tool to predict if this immunotherapy works. because this is a long process, many shots over 3 to 5 years. And so this tolerance, we think, is related to induction of allergen-specific IgG. When we take a serum from patients that have been successfully treated, and we deplete the IgG before we do the assay, then the reactivity comes back to what it was before. So really, during successful immunotherapy, the IgG, specific IgE levels don't go down. They actually often increase, but what increases a lot is the allergen-specific IgG that then neutralizes the allergen and is a protective factor. Good, I'll skip this part for time reasons, and just want to give you 2 short examples of what we're now doing in my research lab. So these are academic projects using this phosphatase system, not for diagnostic purposes, but to understand food allergy and how digestion of food in the gastrointestinal tract is affecting reactivity to food allergens. And this is something, there's many questions. So what happens after digestion of food? Because this is not really known. And so here we had the pleasure to collaborate with Dr. Egger, also from Bern, who is a co-inventor of this so-called InfoGest protocol. So they simulate and standardize gastrointestinal food digestion in vitro. So they take a given amount of food protein and then digest it either. So they prepare an oral phase that is not digested, and they prepare a gastric digestion. So it's the right pH, the right buffer, the right enzymes that are in there, pepsin, lipase, mix it and digest it. And then it goes further into the intestinal phase where they add the chyme, the pancreatin, trypsin, etc. So they really go through the steps of digestion in vitro, and then they analyze the products by mass spectrometry, so what peptides are generated, etc. And they didn't really have much interest in allergy, and we didn't have much experience with in vitro digestion, so we teamed up, and so this is going in a very interesting direction because we get very interesting results. And so what we do here is we don't use patient samples yet, but rather we use a monoclonal IgE antibody specific for a certain food allergy. And so one project we work on is milk allergy, and we have access to a monoclonal IgE against beta-lactoglobulin. So this is a major milk allergen, especially in children that have milk allergy. And so the setup is otherwise the same. So we load the mast cells with this milk allergen-specific IgE, let it overnight, and then we give these fractions from the InfoGest digest, or raw milk, or recombinant beta-lactoglobulin, or cheese, or yogurt, or whatever you want to test, and see what the cells do. And so what we see is highly interesting. So when we test the non-digested oral phase of cow milk in this assay, so these are now sensitized cells with this IgE, we see a response. So the mast cells react to the raw milk proteins, as you would expect, not dramatically, but there's a clear reactivity. But now, upon gastric digestion of the milk proteins, you have a dramatic increase in reactivity. So really, partial digestion by pepsin massively increases the reactivity in the assay. And then when you fully digest the proteins in the intestinal phase into small pieces, 2 to 4, 6 amino acids, then of course you completely lose the reactivity again. And so this is really an early starting point where we want to really explore what kind of peptides are formed and how can we correlate this with eventually patients, and how can we learn from this, what happens. Also, because we're a pharmacology institute, we're also interested in the effect of gastric stomach acid inhibitors, so proton pump inhibitors, which are widely used or prescribed, and by increasing the pH in the gastric fluid, you really affect digestion a lot. And there's some literature that the proton pump inhibitors may increase your susceptibility towards food allergy. And so that's something we're interested in. And the other project, skip that, is the meat allergy. And I have to mention this because you're in Brazil, you eat a lot of meat. And so there is this really meat allergy, red meat allergy called alpha-gal syndrome. And here, very importantly, this is an allergy against an alpha-gal epitope, which is not a protein or peptide epitope, but a sugar epitope. So it's galactose, alpha-1,3 galactose. And so two sugars linked in a way that we humans cannot link anymore. So we can become allergic because we humans have lost the enzyme that makes the alpha-gal epitope. But all other mammals and bacteria present and have this alpha-gal epitope. So this can be seen as foreign by our immune system. So it's very strongly expressed on innards and meat of mammals, either as glycoproteins or glycolipids. And you can become allergic to this red meat or to this alpha-gal by after a tick bite, specific ticks that in their saliva have a lot of alpha-gal and have a strong immunostimulatory modulators, and that can sensitize you towards this type of allergy. And the reason is that we all, or most of us, have alpha-gal specific IgG and IgM, and that's the major problem for tissue rejection upon xenotransplantation. So this is the same epitope, the alpha-gal. It has been long for a long time. And what we think happens, or what is thought happens, is after the tick bite, you induce a class switch from IgG to IgE, and then you become allergic to the alpha-gal. Or you can become allergic to the alpha-gal. One big difference to all other IgE-mediated allergies is that the symptoms don't develop in minutes, but rather hours. So it takes 3 to 6 hours for the symptoms to develop. And that's probably why there's a lot of undiagnosed or wrongly diagnosed cases in the past, because allergologists or doctors never thought of IgE-mediated allergy and rather of an other kind of disease sensitivity. And lastly, the alpha-gal epitope is also present on some biologicals that produced in mammalian cells that express alpha-gal, such as sp2O cells, and one the best example being cetuximab, so the anti-cancer antibody against EGFR, carries alpha-gal, and there have been cases of strong allergic reactions against cetuximab. And the alpha-gal is also similar to the human blood group B antigen. And so if you have blood group B, you might be less susceptible to developing alpha-gal syndrome than the other blood types. There's really a good correlation of this tick, so it's especially the Lone Star tick in the US. And here on top, you have the habitat of the tick, and in blue, the registered alpha-gal syndrome cases, and there's a very, very clear correlation. So where you have a lot of these ticks, you have a lot of new diagnosed alpha-gal syndromes or red meat allergy, as it's called. So we had to modify our HOXB8 assay to assess red meat allergy. And so a question that we can discuss after, or maybe question now to all the students: why did we need to modify this test? And what did we need to modify to make it work? And we spent a lot of time figuring it out, and we made it work, also using a monoclonal IgE antibody against alpha-gal. We got beautiful activation now to alpha-gal sources, including cetuximab, so this monoclonal antibody that unfortunately carries alpha-gal moieties and can activate the mast cells. And just to finish off very briefly, if you're allergic to red meat, you can still eat chicken. And have your chicken heart barbecues if you like that, because chicken are not mammals and they don't express alpha-gal, but all other mammalian sources, especially kidney, intestine, sausage, liver, so the innards are very high in alpha-gal, but also raw beef, hamburgers, and is then really problematic. And also products, you know, gelatin, anything from mammals can be a problem. And so this is the last data slide. When we use this InfoGest digestion protocol, we saw something extremely interesting, or I think it's extremely interesting, is that in the digested preparation, we see a pretty clear activation. It's increasing a bit up on gastric digestion, not as much as for the milk, but now very, very strikingly different on full intestinal digestion, so where you really cleave all the proteins in the little pieces. We dramatically increase their activity towards the sugar, because this alpha-gal is not cleaved in the upper intestine, and it's cleaved much further down or taken up by bacteria. And so there's a massive increase here, and we think this might explain why it takes, you know, some time for the symptoms to develop in these patients, because the food, the meat has to be digested to a certain extent to you know, expose this alpha-gal epitopes. I don't really know why, but there's certainly something that we now work on and try to figure this out. Okay, sorry for having taken so much time, but I'd like to acknowledge people in my lab, especially Daniel Kaufmann, who's done a lot on this, my close collaborator and colleague Alexander Eckel, who really initiated this whole thing at the time together with Noemi Bern, collaborators at the Abroscope, so Charlotte Ecker, Letta Portman with this InfoGest and digestion, the people at the company, Atanis, especially Robin van Brummelen, who's heading the research part of the company, and Jean-Pierre Kina, who generated this knock-in mouse and who's also now actually the CEO of Atanis Biotech. And this is my lab in Bern. I'd like to close here. Thank you very much for listening, and I'm very happy to discuss or take questions if you have some. Thank you very much.
Thank you for this exciting talk. We usually open for the student first if they have questions. So, if you have any questions from the students here. No, I don't know if in São Paulo or online we have questions.
There's one in the chat, Bruno.
Yeah, that's one. I would guess it's Priscilla Olsen. Yeah, she's faculty here. So we can start with Priscilla's question. And that's she says that it was a great lecture, amazing results. Diagnostic accuracy for peanuts is very good. Did you test it for how does mites? I guess was done just before you showed this data. But it seems the response was smaller for HDM. Did you use the whole HDM? And what species of dust mites were used?
Yeah, well, it's a good point. So these are also only really 3 patients for these allergens that we had in our first paper, and we haven't done much more for house dust mite, but we get now stronger responses too. So it really depends also on the quality of your allergen preparation, which can be a big issue. For the house dust mite here, I don't know by heart what we use, but I can certainly let you know. If you can contact me by email if you wish to know. But that makes a big difference. So getting the patient samples is one big part of the work for this clinical validations, and then a good standardized allergen preparation is not always easy to find, and there's big batch-to-batch variation. Then you have to be really careful. You can buy a lot, but not everything works. By the way, I think our Hogsbate assay could also be useful to help to standardize allergen extracts, actually, because just our cells always react the same, so they're pretty stable, pretty solid, and so we could do batch-to-batch comparisons and things like that. But the fair point, I mean, we don't always get 80% activation. It's there's some variation. We're working hard to define really the cutoffs and what's positive, negative. What's clear is negative is pretty flat line, and we have a cutoff for most allergens that we're now developing. Also within the company, the background is always below 3-4%. So that's our background maximum, and a cutoff for the peanut is at around 7-8%, something like that. So anything higher than 7% activation is considered a very positive result. Yeah.
Okay, we actually have another question that was posed in the chat. That's given that IgE-mediated mast cell degranulation is a key driver of anaphylaxis, how might CRISPR-based gene editing to Fc-epsilon-1 subunits in HOXB8-derived mast cells be leveraged to create hypoallergenic or tolerogenic cell models for immunotherapy development in the context of adoptive cell transfer experiments?
The question is if that's feasible, or if it's, I didn't get the first part. How about doing this, or?
Well, it's I guess what she's asking is that if you can actually use these immortalized progenitors for genome editing, like
Yes.
with CRISPR,
Yep.
and then use these cells as different models when you transfer these cells back to the animals.
Yeah, so first part, yes, we can CRISPR out things. Actually, we did. So maybe I can just refer to the AlphaGal question I had to the students. So why did we have to modify the cells? So our cells came from a mouse originally, and so they're actually full of AlphaGal on the surface. And so there was no reactivity whatsoever in the assay initially, because the alpha-gal IgE, that doesn't bind to the receptor, but binds to the alpha-gal epitope, and then zero reactivity. So we had to knock out this gene, this galactosyltransferase gene, by CRISPR. That's the modification, one of the modifications, and the other was that we had to remove all mammalian sources in our culture conditions, so no FCS or no fetal calf serum, because that is full of alpha-gal too, and so we learned the hard way. But certainly it's established to edit these cells or to CRISPR out genes. We do it all the time. About the retransfer into animals, yeah, I think it's an excellent idea. We haven't done anything in that direction so far, but other people have reintroduced HOXB8 cells, and not mast cells, but neutrophils, back into mice, and that seems to work. And so for the mast cells, that should also work because they're pretty long-lived. It's an excellent suggestion, and it's certainly worth for someone interested in hypoallergen development to consider that. Yes, I think technically totally possible. Yes.
We do have I don't know, Gustavo, there is someone next to you, and
yes,
is that a question
there's a
there?
student
Okay.
here that want to ask the question.
Good afternoon. Really nice talk. Thanks for the presentation. And I found it really interesting that the rat mediality took so long to present its symptoms. And I was wondering if you know why this happens.
Well, thank you for the question. It's an excellent question. We don't really know. We can just speculate that this alpha-gal sits at the end of sugar chains. It's always at the top of sugar chains, either on as a form of glycoprotein or glycolipid. So it's surprising that there's no immediate strong reaction. I mean, there is a reaction, right? But that it increases. It's probably due to valency. So if you cut up the proteins into small pieces, you have many more parts, peptide bits that carry a alpha-gal positive sugar chain. So I think you have more epitopes available and a higher chance of activating the receptors probably. But it's at the moment pure speculation. So it's something we want to test now, really. As I said, take these fractions, these digested fractions, and then do a mass spec analysis, see what peptides we form. It might have to do with, a lot of food initially is present in the form of mice cells. It maybe not be accessible. And so there's many open questions, and we ask, exactly the same question as you, but we don't have an answer. Yeah.
Okay, thank you. We have a question here from Miriam.
Hi, thank you very much for your talks. Really, really interesting and a lot of food for thought there. So I had a question and a comment, and the question was related to the peanut allergy lines or graphs of response that you got, and you said that you get 10 to 15% of allergic people that fall below the detection line. And I wonder if you think that could be just a number that you have, or if could be related to the sample handling, or maybe the age of the patient. You said that sometimes you get pediatric cases.
So, well, thank you. I probably wasn't clear then. So thank you for giving me the chance to correct myself. So we don't have 10-15% below the threshold. It's the 10-15% of non-responders in the basophil activation test. So when you do the basophil activation test, you take fresh blood from a patient, you add the allergen and the staining antibody, and you want to activate the basophils in the blood. And it's very similar to what we do, but with the patient basophils in fresh blood. And that test works beautifully, except that you need fresh blood and you cannot store it, etc. But it works very well. But 10 to 15% of the population will not react in this basophil activation test because of intrinsic issue with the basophils of the patient. And they're called non-responders, and they could be allergic patients, but their basophils don't respond, but the mast cells will react. And these non-responders are usually, I hope I'm not, offending anyone who's doing these studies, but often they are excluded from publications or from studies because they don't respond. And we wanted then to test that in our assay with the standardized cell line that is not from the patient, which is a caveat in itself, but it's also an advantage because of, they're always the same cells. We don't have this issue. So blood from non-responders worked well in our assay, so we could solve this. Having said that, these were only a handful of patients we tested so far, but we're pretty confident. We don't see a reason why this shouldn't work in our assay, because we only use the serum part of the patient and not the cell intrinsic.
Okay, no, that's really interesting, because it seems like a very powerful system also to test the effectiveness of several vaccination protocols, right? You could test any antigens. And so the comment that I was going to make, I found it very intriguing that after the tick bite, you changed and you became allergic. And I've recently heard of a couple of cases of people who got food a gastrointestinal infection, and after that became intolerant to milk. and it's not known if it's lactose or if it's protein based, but reminded me as well as a paper from 2019 from Musida's lab, where they saw that you have compartmentalization of the intestine lymph node chain. Depending, there's a set that reacts more to the beginning of the intestines, and another set to the later parts and the colon. And they have also different reactivity to protein or to sugar. So it all kind of comes together. And I wonder if you saw or if you discussed any cases of also acquired milk intolerance due to infection, and if that could be related to something as you saw for the tick.
Oh, that's very intriguing. But no, we don't. Most of what we do, like in my lab, is we don't work so much with patient samples for research because they're hard to get. But more with monoclonals or mixes of monoclonals, which are also not for each allergen easy to get. But it's a very intriguing question. Yeah, there could certainly be more to it. And for the tick, you could think of, it's not only this Lone Star tick, there's about 20 tick species that can do the same. And then so there might be, might not only be true for the alpha-gal, I don't know. So it could be much more, but it's pure speculation again.
Right. So before I think we have another question there in São Paulo.
Yeah. Yes.
Great.
Thank you for the nice talk. I was wondering if your team intend to integrate organs on chip on this approach to evaluate the possible efficacies of types of therapy to induce tolerance and the effects on a more systematic way, or to analyze the effects on the tissue specific?
Great question. Not at the moment. I mean, that's way beyond my expertise, but of course, sounds fascinating if someone has the expertise. Of course, we're very open to for collaborations, but it's not something that we're following up. So we're much more at the basic research parts, trying to understand very basic things. But yeah, of course, eventually that would be fantastic. And I also have to mention the caveats of our system. It works well, but there might be these are in vitro differentiated mast cells. I think they're similar to the bone marrow derived mast cells that people use in the research, but there's subtypes of mast cells in vivo or different kind of mast cells that can be quite different. So that's certainly very, next step or even after to go into these differences. But yeah, it's a great question again, but no, we don't do that at the moment.
Okay. I was wondering if the human Fc-epsilon-R1 in these mast cells, they actually increase the membrane presence with IgE, if IgE stabilize them. And if you ever try to put IgE when you start the mast cell differentiation with the withdrawal of tamoxifen, and if it makes any difference in the mast cell response to the allergen afterwards.
Fantastic question, Bruno. Great. Yes, absolutely increases. So it stabilizes the receptor on the surface 2 or 3-fold. So that's why when we do this passive sensitization, we always do it overnight. That works much better than doing it. I mean, it binds within minutes. I mean, after 1-2 hours, all the IgE is bound to the mast cells, but the response is much better if you leave it overnight. And that's exactly the reason, because you increase the receptor density on the surface, because the new receptors that bind the Ig on the surface are not recycled, are not very fast. So you increase. And so we also, during this sensitization, we increase the IL-3 concentration quite a bit. So this, we call that activation medium. And the IL-3 also helps to prepare or prime the mast cells. We've played around a bit by adding Ig from the differentiation start, you know. There's not so much difference in the end. So 1 day is enough in our hands. So we do differentiate the cells for 5 days, and then we put the IgE for 1 more day with high IL-3, and that's, I think, is the maximum response we can get.
Okay,
We
great.
have, you know, there could be other cytokines to play with, or yeah, you know, we tried
mast
a
cells
couple.
can get messy, right? With all the different types, different tissues, and everything. We had raised hands from Lucas Bolini, but I guess he just put his hand down when I asked my question. So I believe I asked his question, but oh no, he actually put here on the chat now. I was going to ask how well HOX-B8 mast cells can be added back to mice, and whether you checked whether your mast cells exhibited a phenotype that resembled more mucosal or connective tissue mast cells. But I get it's ongoing work. Thanks for the talk. But do you have any comments on what is the phenotype of these mast cells that you generate, and if you can add them
Well,
back?
yeah, so I think, again, they're pretty much the same as bone marrow derived mast cells that you prepare with IL-3. And so they're not really specialized mast cells, but they resemble more the mucosal than the connective tissue, I think. But you know, they're a model kind of mast cell. You, of course, could tweak it with adding specific cytokines to make them more connective tissue-like, more mucosal-like, but it's not something we have really done yet. And we also haven't put the cells back into mice. This has been done. We did it with the basophils a long time ago. So we had the GFP labeled basophils, and that was tricky. We didn't really find them so easily. So the problem is they're not super long-lived. So that's a big difference to primary mast cells or basophils. So when you differentiate them, they live for, you know, they slowly die off, and then you can keep them alive. And then we have, you know, we can keep them alive for 2 weeks or 1 week maximum. But for our assay, it's enough, because as long as you have a solid population that is viable, the assay works nicely. So it doesn't really matter if you have dead cells around, so you get on the viable cells. So we're happy when we have 40-50% of viable cells in the assay. That's good enough, but to put back into mice, that is not good enough. That would need some optimization and some, you know, some more work. Again, this has been done for other HOX-B8 lines by others, neutrophils, and there's publications that this works, but then you have to be pretty quick doing your experiments, you know, within a couple of days. Yeah. So I think we're not quite there yet to really put them back and then follow up long term. Yeah. Sorry, what we haven't done, and you just gave me an idea maybe, so we have never put back the progenitors. I'm not sure anybody has done that. So people have just put back differentiated cells, but it might be interesting to put back the progenitors and see what happens.
Definitely. Okay, do we have any more questions? People in the room, people at USP?
No, here everything.
Okay.
Everybody's quiet now. Just want
Sure.
to thank Tom again. It's great to see you. Hope
Pleasure.
that Bruno and I will manage in the future to bring you here. So it's
Sure.
this is a scene. You haven't been to Brazil, so we got to do something. And yeah, if I'm lucky enough, then on September we'll meet in Bern.
Yeah, that would be fantastic.
All
I
right.
will send you the flyer. So if anybody's interested in cell death,
Yeah,
wants to come to
definitely.
Switzerland.
Yeah,
Cool.
send us and then we'll advertise.
Fantastic. Well, thank you very much.
All right. Thank you. It was a really exciting talk in a very unique theme. You know, there are not that many people, as you know, working with mast cells and particular basophils, and you have been doing a really a very exciting work. And it's also very nice to see things that are done at the lab bench, getting to the patients and improving lives. So it's congratulations on all of your work and on this very nice talk. Thank you.
Well, thank you, Stukan. Thank you. And sorry for being so long. This is the older I get, the more I talk because
All right. Thank you. Bye bye.
Well,